My name is Caleb Dunn, and today we're going to be talking about the structure of antibodies and the different types of antibodies that are found in the body. So an antibody is a specialized protein that will travel throughout the body, and it's going to be found in different bodily fluids, and they'll travel through the immune system and help to defend against foreign pathogens in the body. And these antibodies are produced by a B cell, which will develop from stem cells in the bone marrow. And when these B cells become activated by the presence of an antigen, they will develop in the plasma cells. And these plasma cells are what generate the antibodies in the immune system. So these cells consist of 4 different chains, which are 2 light chains, which will be represented by the color green, and 2 heavy chains, which will be the color blue. And these are going to form a Y shape. And each light chain in the antibody is going to be bound to its partner heavy chain, and this is going to be formed by a disulfide bond, which will be represented in black, and that's going to link these two together. And both of the light chains and both heavy chains are going to be identical to each other. And about halfway down the length of the antibody, there's going to be multiple disulfide bonds, which will be about right here, that will link the 2 heavy chains together. And the outside part of the antibody is called an antigen binding site. This is the antigen binding site. And below that is going to be a region called the crystallizable fragment region, which is also known as the FC region. So the antigen binding site region is going to recognize other antigens, similar to how the receptors of B and T cells, they'll recognize pathogens and clonal expansion. And the crystallizable fragment site, they'll interact with other parts of the immune system. And because each antibody can recognize different antigens, the antigen binding sites can be different for certain antibodies. And this region of variability is known as the variable region. And just below that, in the variable region, which will be about right here, this will be called the hinge region. And this is going to be located around the disulfide bonds, and it's going to give the antibody flexibility, which will be helpful for when the antibody wants to bind to an antigen. So there's 2 major classes of antibody light chains, and these 2 classes are variable light and constant light regions, which will be represented by the Greek letters kappa and lambda. And the type of heavy chain on an antibody will determine the class of an antibody, which are also known as immunoglobulins. So the 5 types of antibodies are going to be IgG, IgE, IgD, IgA, and IgM. And the first class of antibody that we'll talk about, IgA, this has an alpha heavy chain, and it's going to be found in the mucus, your saliva, your tears, and your respiratory and gastrointestinal tract. So it has a little bit of a different shape than some of the other antibodies. So the heavy chains will start out this way, but then they'll actually be formed together this way, and your light chains will be out here. And then your disulfide bond will be linking them together this way. So a little bit of a difference between the IgA molecule and the rest of them is that represented by red, they have what's called a secretory component. And what this does is it's going to protect them from being broken down by some of the gastric acids in the digestive system, because they're found in your digestive system and also your respiratory system. And they're going to protect against different pathogens invading the body. So this is an IgA molecule. Your second one is going to be IgD, and it has a delta heavy chain, and it's going to be part of the B cell receptor. So your light chains will be out here, and your heavy chains will be like this. And here are your disulfide bonds. But down here in the heavy chain, they'll only have one disulfide bond. And what this antibody does is it will activate basophils and mast cells in your body. And your third type of antibody is IgE. And this is represented by the heavy chain epsilon, and it can be found in your mucous membranes and your skin. So again, here's your light and your heavy chains, your disulfide bonds, and unlike D, they'll actually have up to 3 or 4 disulfide bonds. And this is going to protect against parasitic worms, and it's going to be responsible for some of the allergic reactions that your body has. And the fourth is IgG, and this is the most common antibody that will be found in your body, and this is represented by the heavy chain gamma, and it's secreted by plasma cells in the blood. So again, here's what this looks like. You got your light chains out and your heavy chains. And this will have 2 disulfide bonds, which will be the type of antibody that I drew up here. This is an IgG molecule, and this type of antibody is going to be able to cross the placenta into a fetus, and it's the most plentiful antibody in circulation. And the final antibody, and probably the most complex, is the IgM molecule. And this is going to be represented by the heavy chain mu, and it may be attached to the surface of a B cell or secreted into the blood. So this type of antibody is going to have a bit of a different shape than some of the others, as their heavy chains are going to look like this. this isn't the best drawing, but it'll have a type of pentagon shape with the disulfide bonds, and out here, of course, you'll have your light chains as well. And your extra disulfide bonds linking the light and heavy chains together. So this antibody is going to be responsible for the early stages of immunity in the body, and there's also a few subclasses of immunoglobulins that are based on the differences in the heavy chain units of antibodies. And there are also specific types of antibodies called monoclonal antibodies, and they're produced by a single clone of cells, which will mean they're a single type of antibody. And they can be produced in large amounts in a laboratory, and they can be useful in fighting against cancer. And that's the structure of antibodies and the different types of antibodies that are found in the immune system.